Good afternoon to everyone. Today I'm going to take Streptococcus pyogenes, clinical manifestations and laboratory diagnosis of Streptococcus pyogenes. You all of them already know about Streptococcus is a gram-positive organism arranged in the form of chains. And this is the first classification. First, I'm talking about the classification of Streptococcus. Classification is based on the oxygen requirement. The streptococcus are classified into aerobes and anaerobes. Aerobes are again classified based on the growth on blood agar medium. They are alpha hemolysis, beta hemolysis, and gamma hemolysis. And anaerobes, only one example, that is the streptococci. And depends upon the growth on the blood agar medium. They are classified alpha hemolysis. Alpha hemolysis means incomplete hemolysis. Example is Streptococcus viridans. And beta hemolysis means there is a complete hemolysis are shot by this organism. And then again, it will be grouping, Landsfield grouping, that is based on the carbohydrate C antigen present in the cell wall. And again, it will be typing also came, that is the Griffith typing, MTR, that is depends upon the MTR proteins. Then gamma hemolysis, and here in beta hemolysis, the one example that is Streptococcus pyogenes, and alpha hemolysis example is Enterococcus faecalis. Next, directly we'll go with the infections. Organism, it's a gram-positive organ. I already mentioned that it's a gram-positive organism arranged in the form of a chains. It's a non-motile organism, and organism having the capsule, hyaluronic capsule are present there in the organism, and it's a non-sporulating organism. Then important mediums are used for the cultivation, that is blood agar medium, and then we can use all the enriched medium for their cultivation. In blood agar medium, organism will show the hemolytic property. Then we'll, with the infections, organism have a lot of capability to produce antigens, enzymes, and proteins. These all of them have the contribution to virulence in case of Streptococcus pyogenes. Antigens are group C carbohydrate antigens, protein antigens, and then capsular antigens are everything are present in Streptococcus pyogenes. The main infections are produced by the streptococcus are they are separative infections and non-separative infections. The separative infections are mainly respiratory tract infections. In the respiratory tract infections are sore throat, tonsillitis, pharyngitis, scarlet fever, and pneumonia. And skin infections are the erysipelas, impetigo, lymphangitis, cellulitis, pyoderma, wound infection, burn infection, and necrotizing fasciitis. And all these infections, organisms are produces a lot of virulence factors. Then enzymes are producing and other antigens are producing. And these have the contribution to virulence also. The important toxins are produced by these organisms are hemolysins and O hemolysin and S hemolysin. O hemolysin is responsible for the diagnosis of streptococcus, and S hemolysins are present in hemolytic. They will be associated with the hemolytic property on the blood agar medium. The first one, the skin infections. Skin infections, the important in the separative infection means the organism is a pyogenic cocci, and pus-producing organism. The main skin infections are erysipelas. Erysipelas is an infection of the upper layers of the skin. It's a superficial infection, and it results in the fiery red rash with the raised edges. Then impetigo. Impetigo is another important skin infection. It is common and highly contagious skin infection. Mainly affects infants and in children and appears as a reddish sore on the face. And it usually will tell us honey-colored crust. Sores are burst and develops. It's like a honey-colored crust that you can make out there in the figure only. Then it will be especially seen around the mouth and on the hands and feet. Genital infections. Other infections are the genital infections, purpural fever, purpural sepsis, and other infections are the otitis media, mastoiditis, peritonsillar abscesses, suppurative cervical lymphadenitis, Ludwig's angina, meningitis, septicemia, pyemia, endocarditis, and abscesses in internal organs. The next one is a non-separative complications are also there. Non-separative complications are mainly acute rheumatic fever and acute glomerulonephritis. I will explain later. Then first we'll explain with a respiratory tract infection. I already explained the skin and soft tissue infections. That is the important one is erysipelas and impetigo. Then next one is respiratory tract infections are tonsillitis. Most of them are know about the tonsillitis. Sore throat is because of the Streptococcus pyogenes. That is the important disease is caused by the Streptococcus. And then tonsillitis, inflammation of the tonsils. And then other one, it's already discussed about the erysipelas and then impetigo that you can make out here. The next one, the wound infections, burn infections, important infections of the skin and soft tissue infections are the wound infections and burn infections. And here, non-separative complications, just already I named as acute rheumatic fever and acute glomerulonephritis. I mentioned that in the previous slide. Then acute rheumatic fever is a fever is involving hurt and joints. That is the antigen cross reaction between the group A streptococci and myocardium. And acute glomerulonephritis, that is the cross reaction between the antibodies are produced against the streptococci and antigen of glomerular basement membrane or deposition of antigen antibody complexes. And the acute rheumatic fever, the principle behind this is antigen cross reaction between streptococci and myocardium. And glomerular nephritis also the same thing, the antigen cross reaction between the streptococci and glomerular basement membrane. The next one will go with the laboratory diagnosis. And before that, I will explain once more. I will tell about the infections. The main infections are caused by the streptococcus are skin and soft tissue infections and respiratory infections and other infections and genital infections. Then respiratory infections are mainly tonsillitis, pharyngitis, and other meningitis, and other complications are also pneumonia and other complications are also there. And the skin and soft tissue infections are important. One is erysipelas, impetigo, burn infection, wound infection, and the other infection, general infections are purpural sepsis and purpural fever. That is mainly because of the contamination is coming from the hospitals. And laboratory diagnosis, the important specimen is blood, pus, CSF, throat swab, and vaginal swab. In the blood, pus, CSF, throat swab, and vaginal swab. All of them know about the specimen collection is depends upon the infections and transport. First, you already collect the sample and transport with the Pike's medium. Pike's medium is an important transporting medium using for Streptococcus pyogenes. And then the first examination, it's a microscopic examination. Organism, it's a gram-positive organism arranged in the form of a chains. That is a microscopic examination. We have to go for, we have to do with the gram staining. And the next one, cultivation. In the cultivation, do with different medium. Different medium, the important medium are used for the cultivation is enriched medium, blood agar medium. Organism produces beta hemolytic colonies. Organism produces beta hemolytic colonies on the blood agar medium. Colonies are small and smooth colonies, smooth circular colonies. And biochemical reaction, organism fermented some sugars and it does not ferment ribose and sugars and produces acid. It's a catalyzed negative organism because streptococcus is a catalyzed negative and staphylococcus is a catalyzed positive organism. PYR test is a positive test for streptococcus. Pyrrolinodyl naphthalamide test is a important test for the biochemical reaction. And then antibiotic sensitivity, we have to first we'll go with the first we have to collect the specimens, transport it. Next we'll go with the microscopic examination. Next we'll go with the cultivation, and the next one with the biochemical reaction. Next test is antibiotic sensitivity test. How many antibiotics are showing resistance or sensitive for this. The next one is antigen detection, and then nucleic acid detection. Nucleic acid detection will be using for most of the organism for their nucleic acid by PCR test. Then antibody detection by streptosome test, passive agglutination test. It's a passive agglutination test, and ASO test. ASO test is the important test used for the diagnosis of Streptococcus pyogenes. ASO, anti-streptolysin O. Anti-streptolysin O, it's have the antigen. O antigen have the antigenic property, and more than 200 will come. It's a positive result. Then the last one, that is the treatment. The important treatment is penicillin G, erythromycin, and cephalosporins. And this is the actually I want to discuss in this today's class. I want to discuss about the infections. What are the infections, and then what is the laboratory diagnosis and treatment of Streptococcus pyogenes. Thank you.